Welcome. My name's Rod Chalk. This is the third in our mass spectrometry training seminar series, and today we'll be learning, this is the only practical in the series, and we will be learning about how to use the Mascot server in order to search MSMS data sets. So hopefully everyone will have some files that can be searched. Either that you have your own files or there's files that I've sent you. These are .mgf files. I sent them in the last email but one. If anybody hasn't got those files, I would like everyone to take part. So if you haven't got those files, can you unmute, let me know, and I will send them to you now. And because this is a practical session, I would suggest that everyone stays muted. But if you would like to, enable video, that will help me. If you have any problems with what we're trying to do, you can wave at me, and hopefully I'll be able to help you. We will be using the Matrix Science web server. What I would suggest is that we split screens so that you can see what I'm doing and you can see what you're doing at the same time. If anybody has data that they would like me to analyze using this server, I didn't hear anything from anyone, so I'll assume that there isn't anyone who wants to do that. So we will be using the 6.mgf files that I've sent you. Now, for future reference, if you send any samples to a mass spec lab, most labs will be able to provide you with data, MSMS data in this format, in .mgf format. It is pretty much standard. The Mascot server will accept other formats as well, but this one I find is the easiest one to work with. So just to remind everybody, the session is being recorded. I would ask for this session that if anybody has any questions, they unmute and they ask them as we go through the session. I'm quite happy, there's not too many of us, so I'm quite happy that that should work. And I think the important thing is that I answer questions as they come up. And I think we shall make a start. So I'm going to share screens with you. And what I would like you to do is to go to your web browser and search for Matrix Science. Okay, so this is Matrix Science, is the company that developed Mascot. And if you click on that, you should get to the Matrix Science web page. So what we're going to do is we're going to do a Mascot database search. So we're going to highlight this. And we're going to access the Mascot server. And if I just scroll down, there's a number of different searches you want. Peptide mass fingerprinting is not used very much. This is the one that we want. MSMS ion searches. And if you can see, the one that I've highlighted, perform search. So that will bring us onto the MSMS ion search page. So if you're going to follow me with this, you need to put your name in. You are also going to need to give Matrix Science your email address. They're not going to spam you. This is so that they don't get the server swamped by bots. So you need to put that email address in. The first time you attempt to search, it will come up with an error. It will want you to verify that email address. I can't show you that because it's already done it for mine. But when we submit our search, it will ask you to verify that email address before, and then you'll have to submit the search again. Okay, so this is the dialog box. You can add a search title if you want to, it doesn't matter. Let's just call it test. First thing we have to do is select a database. And Mascot has already recommended a database, and this is contaminants. These are proteins that are commonly found in laboratories. So the type of thing that you will find are things like keratin, BSA, lysozyme, trypsin, typical proteins that will just come up randomly in any laboratory. And there's about 200 of these, and this is one of the Mascot databases. We're going to select a database that we want to search. I'm not going to use it, but this one, CRAP. This is known as the crapome, and this is not commonly found contaminants. These are superabundant proteins. So if you are if you're doing a proteomics experiment, you will find that there are some proteins which are always massively overrepresented. Typically, serum albumin, antibodies. There are a large number of proteins that are very, very abundant in the cell or wherever your sample comes from. And usually they are not of interest, but you can search that. The database that we're going to use today is this one, SwissProt. So SwissProt is a curated database. It has about 600,000 entries. Most proteins that are being used for research will be in the SwissProt database. If you're working on something esoteric, it won't be, but it's pretty good for our purposes. And so I'm going to load that. So the two databases we're going to search are contaminants and SwissProt. If you have your own Mascot server, you can create your own databases. There are a number of databases available. There's some databases for model organisms here, which are quite useful. So this server is basically a marketing tool for matrix science to get you hooked on this type of thing. If you do a lot of this, you will find you need to have your own server. Okay, so we can select taxonomy within the database if we wish to. In a lot of labs, that's normal. They will narrow down the taxonomy to human or whatever it is they're working on. Personally, I prefer not to do that. If you keep the taxonomy as everything, you can use statistics to help you work out the significance of your data. If you narrow the database down, you are spoiling that a little bit. We need to select an enzyme. We have a choice of enzymes. Some of these are specific, like trypsin, chymotrypsin. Some of them are non-specific. They've moved it. No cleave, I think that actually means no enzyme. But we'll stick with trypsin for now because trypsin is the most commonly used enzyme. This is quite important. It's quite normal for trypsin or any enzyme you use for digestion to be incomplete. It's absolutely normal, particularly in a proteomics experiment, and so you must allow for a number of missed cleavages. On this server, you can allow for 3 without any problem. But there is an issue with this, is the more missed cleavages you allow for, the more complex the database search becomes and the more CPU time it uses. If you have a very large database and you allow for a very large number of missed cleavages, this server will simply not allow you to do it. It will kick you off. If you have your own server, you may be able to do more of these. On our server, we would typically use 4 missed cleavages, but the more missed cleavages you can add, generally the better. We're not going to use the quantitation or cross-linking functions today. What we are going to use is we're going to use these modifications. So normally, LC-MSMS data sets will have fixed modification, and the normal modification is this one, carbamidomethyl cysteine. So all the cysteines have been reduced and then they've been alkylated. So they will all have that modification, and that's a global modification. So every single cysteine in your sample should be modified with this modification, and there's a mass shift associated with that. Now, the second type of modification we can specify is a variable modification. And for a normal data set, the only one I would recommend is oxidation of methionine, which is there. You can see alongside that, you have phosphorylation of serine and threonine, and phosphorylation of tyrosine. You can add these if you want, but a word of caution is the more modifications you allow for in your search, the greater number of false positives you will generate. If you include these modifications in a random data set, you will find Mascot generating phosphorylations where actually none exist. So do not use these modification functions unless you have good reason to believe there might be these modifications present. So if you are working on a kinase, by all means include these. If you have enriched for phosphorylation, by all means include these. But for normal samples, the fewer modifications you have, the fewer false positives you will get. We now need to specify our mass tolerance. The data sets I've given you are ion trap data sets, so the mass tolerance is quite large. I'm going to specify here 1.4 Daltons for the precursor mass tolerance and 0.4 for the MSMS mass tolerance. That's in Daltons. You can change this to ppm, and this would vary depending on what instrument you're using that your MSMS data came from. If it's a TOF instrument, it might be 10 ppm. If it's an Orbitrap instrument, it might be even less than that. We're going to specify, we can specify carbon-13 isotopes, so we can specify one. So it's possible that one carbon-13 isotope could be fragmented. We need to specify which charge states we're going to look for. My preference is for this, +1, +2, +3. For a peptide data set, you could alternatively go miss out the singly charged and go +2, +3, or +2, +4. But I will stick with this one. For when we've got an option of monoisotopic mass or average mass. When you are searching peptide data sets, you must always use the monoisotopic mass. Okay, average mass is only for large MZ species such as proteins. We're now going to select our data file. So I'm going to browse here, and I'm going to load the first unknown protein. It's not unknown to me, I know what that is, but you don't, so we'll load that. The data format is Mascot generic, I've already mentioned, that's .mgf. There are other formats here. These are specific to a particular type of instrument. If that's the format you've got, you should be able to search that as well. You have to specify the instrument. Okay, different instruments produce different types of fragments, and this instrument is electrospray trap. So we will select that for the fragments. We'll include a decoy database search, and then I'm going to start my search. So when most of you, if you press this, you will get an error asking you to verify your email address. So I'm going to press this now, and it's going to start the search. So my search is running. For you, what you will have to do is wait until you get the notification email. It might take a few minutes. and then run that search again. Are there any questions while we're waiting for the search to complete? If you have any questions, please unmute and ask them now.
Can you speak just a little bit about the tolerances, like what that actually represents?
Yes. So whatever mass spectrometer you use, it will measure the mass to within a given tolerance. So typical tolerance for an accurate mass instrument would be, say, 10 parts per million. So that would be for, say, if you had a protein with a mass of 100,000, then that would be work it out, it'd be about 10 Daltons. So the tolerance is a measure of the type of instrument it is. You can have different types of instruments. So high mass accuracy instrument would be something like an Orbitrap. I would expect a mass tolerance of about 1 part per million. For a time of flight instrument, 5 to 10 parts per million. For an ion trap instrument, it's much lower than that. It's about 300 parts per million. And we specify that in Daltons rather than in ppm. However, that's not really the important thing. The important thing is the fragmentation match, as I'll show you in a minute. Does that answer your question?
Yes, thank you so much.
Okay. So we have some data. It's returned a result for us, and it's listed 2 hits. Different versions of Mascot can do this in different way. This has just listed the hits which it considers to be significant. Earlier versions of Mascot will give you a hit list, and it will just rank the matches to the database. And you must be careful with that hit list, because just because something is on that list or because it is at the top of that list does not mean it's significant. The significance measure for Mascot is down here in the Mascot score histogram. And you can see there's this green hatched area, and this is round about MAUS score 50. And in general, if the score is below 50, Mascot will generally consider it to be not statistically significant. And if it's above 50, it will consider it to be statistically significant. And these 2 hits that we have with a MAUS score of roughly 200 and I think 420, these are very, very highly significant. We also searched a decoy database. Now, the decoy database is, I don't know exactly how Mascot construct this database, but basically it's a random database. So it's junk sequence data. And our data set has been searched against that junk sequence data, and it has generated no hits. So our false discovery rate is 0. So Mascot has 2 different ways of displaying the data. You can display in this way. This is the peptide summary. This is the method I am most used to, which is nice. There is also a protein family summary, and that is useful, and I'll show you that in one of the later samples. So what we have here is Mascot has identified 2 hits. Okay, so the first hit, this is from the database. This is a spike from COVID-19. This is important. This is the score. This is the Mascot, the MAUS score. So this is a measure of the statistical probability of the match that we have being completely random. And what that MAUS score of 421 means is that the probability, the p-value for this match is 10 to the minus 42 42nd power. So a score of 421 is massively, massively, massively significant. It cannot be arrived at by chance. You have to take off the effect of the database. So the database is large. The database is about 600,000 entries, but even taking that into account, that will still give you a p-value of 10 to the minus 36th power. So it's still massively significant. Interestingly, so we can drill down into this data. Just to tell you, if you're doing this on the server, once you have submitted your search, you've looked at the data, when you exit the server, that search is lost. It's not saved for you. If you do that on the Central Proteomics Facility server, if you do it on the CMD server, we will save that search. So that's a reason for, again, reason for having your own server. So we can click on this, and now it is showing us the protein sequence with our peptides mapped onto the sequence. And the first thing you can spot is actually the coverage for this protein is very low. It's only 6% coverage. However, the Mouse score is extremely high. So do not use protein coverage as an indication of significance. It isn't. The reason that this protein coverage is low is because the protein is quite large. Also, as you might know, the spike protein in COVID-19 is very, very heavily glycosylated. We don't know the mass of those glycans, so we can't pull them out of the database. Also, we didn't deglycosylate this sample, so the glycosylations will prevent trypsin from accessing it. So that's why the coverage is low, but the Mouse score is absolutely fine for us to identify this protein, which was the object of the exercise. Now, you can see that Mascot has identified 6 peptides. And if you mouse over, don't think it's going to, it's not going to do it for me. Not going to do it for me. I don't know why. Normally, if you mouse over this area here, it will show you a pop-up box. I think probably I've got pop-up box disabled, but it will show you the ranking for this peptide that begins TQL, and it will have a score, and that the top score will be 56, but it will then score everything else in the database. Every single tryptic peptide in the database, there are about 600,000 of them, and they're all given a score. And all of those, apart from this one, will be, most of the scores will be 30 or less, and they will be junk data. This score is significant. How do I know that? Well, let's have a look at it. The first thing you want to look at is the delta mass. So this is the difference between the observed mass for this peptide and the calculated mass for this peptide sequence. And that's what you can see here is that they're all kind of round about the same. This is acceptable for ion trap data. It would not be acceptable for Orbitrap data, but it's acceptable. And this indicates that your all of these precursors are real. If we now click on that, not mouse over, but click on query 176, and this will actually show us the real data. So this is our fragmentation spectrum, and if you attended last week's seminar, you'll know what this is. This is the ion table for this theoretical sequence, and where we have a match between the peaks in the spectrum and the theoretical masses in the table, it is highlighted in red. And this number here is calculated from the number of bold matches, the bold red matches. So this one doesn't count, but these matches are used by the Mascot algorithm to generate that score. And a score of 56 is significant. You can see it's less than 0.05. It's a significant score. On its own, that single peptide score would not necessarily be taken as identity. If you had this result and you wanted to publish it, it would not be acceptable. You would need to have at least two peptide scores, two separate peptides from the same protein. We can look at the, if you can see this, yes, you can see. So these are the mass tolerances here. This is for this one, it's 72 parts per million, which is okay for this data set. And all of these values are plotted here. So the mass errors are plotted in terms of Dalton, and then the same data is plotted in terms of parts per million. And what you want to see, if this data set is real, you would expect that the mass errors are small and that they follow a trend. And they do. Almost all of them are fitting on this line. That one possibly you wouldn't accept, but all the rest of them, this is a non-random distribution of mass errors. So that's good. So that is a reasonable peptide. Let's look. I'm going to get lost here now. I may need to reshare my screen. Any questions while we are covering this? Okay, let's look at a higher score. Let's look at this one here, 169. So this is a much higher score. And you can see from this that almost all of the peaks in our spectrum are represented in the table. So nearly everything in here is labeled red. And this is a very good match. So we've got an iron score of 93. That is highly significant. I would, if I had this peptide on its own.
Anyone else not see your screen? I can't see anything.
Oh, okay, that's my fault. We're not sharing. I'm going to go back. I apologize. Let's go back and share screen. Okay, can you see my screen now?
Yes, thank you.
Yep, sorry about that. Okay, what we're going to do now is going to look at this high scoring query 169. Okay, so what you can see here, I'll just repeat myself, is there are most of the peaks in this spectrum are annotated red, and that means they match the ions in the table. So the most significant ions for peptide matching are B ions and Y ions. And this is a very nice data set. We have a continuous run of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 B and Y ions. And the chances of that being random are 1 over 20, because there are 20 amino acids, 1 over 20 to the 12th power. So that is highly significant. That gives us our Mayo score of 93. And if you have a look at the mass errors, again, the mass errors are very convincing. Okay, so that is nice data. I'm having problems getting back to where I was, so I'm going to have to stop sharing again. Apologize for this. let's try and search something else now. So I'm going to reshare screens again. Okay, you should be able to see now the Mascot search parameter screen. Okay, hopefully you can see that. Can I ask, have you all been able to follow this with your own data? Has anyone not been able to do this? Okay, I will take silence as acquiescence. Let's try and load something else now. So we can stick with these these parameters. These are good for all of this data set, and we're going to now load a new protein. It's online protein 2, and I'm going to start search again. Any questions while this search is running? So just to say, Mascot searching, with the data set like this, it can be quite fast. It's quite fast on the Mascot server. The more, as I said, the larger database you choose to search, the more miscleavages you add, the more modifications you want to look for, the longer the search will take. If you're sharing a search, a Mascot server, sometimes it will slow down to a dead stop because somebody has done an extremely large search. So that appears to have finished. Maybe we can refresh it. The Mascot web server won't let that happen. As I said, if the search parameters, you're going to use up too much memory, it will just kick the search out. Doesn't seem to want to show us anybody. Would anybody else like to share their screen? Mine seems to have not been doing very much. Okay, no response to that. And try and refresh this.
I'm getting an error message saying the server's busy.
Ah, yes, probably it's because somebody's running a training seminar.
Thought that might be it.
I did wonder how the server was going to. I mean, it should be used by thousands of people all the time, so I wouldn't have thought we would make that much difference. Has anybody got a result? Is anybody been following me on the second sample and got a result that they can share with us?
Yeah, I'm following, but then in my screen it also shows 99% search running.
Okay. Didn't anticipate this. I think probably there's not too much we can do about it. Has anybody got any questions that we can answer while we're waiting for Mascot to do something?
Is there any way that we can kill all of our searches to allow yours to go through quicker?
I don't know the answer to that. So we don't normally use this server because we use our internal server. Wondering if I can use our internal server instead. Not sure about that. Okay, oh no, it's done it. It's done mine. Okay, that's fine. Mine's done it. I don't know if anybody else has. Can you see my screen? I've now got Mascot search results.
Yeah, I can see.
Unknown protein 2. Okay, so I said that Mascot has two different ways of reporting the data. So the previous way we had a look at was the peptide summary. This is the protein family summary. And that's quite useful because if you have something like tubulin, this is a highly conserved protein. I think we've got tubulin in one of the samples later. It's highly conserved protein, and it's virtually identical from one species to another, which means that your MSMS ion searches, your peptide matches, are the same amongst a whole load of entries in the database. And what Mascot will simply do is it will just list hundreds and hundreds of species that happen to have tubulin, which is just about everything. And the other more interesting parts of your data can be lost too far down the list. So what Mascot introduced some years ago is this proteins family layout, where basically it doesn't show you the matches, it shows you the protein families that match. So this is the layout. You can expand this. So what we have here, this is TEV protease. So this is a common thing that we see in the lab, and we've got a score of 220. That's highly significant. Just to say, this score here, how that's arrived at, is Mascot generates a score for each peptide, and then the non-redundant peptides. So these are all non-redundant, so they're no matching peptides. That is then summed together to reach this score. Okay, so you generally need several peptides to have a good score in Mascot. The other thing that's quite useful is this. EMPAI is some quantitative information. So what it's done is it's taken the intensity for these peptides and compared it to the intensity for peptides from known amounts of a BSA digest. And so it's not a particularly accurate quantitation, but it's better than nothing, and you kind of get it for free because you haven't had to use any special reagents to get quantitative data. It's useful, but not totally reliable. So we've got TEV protease here. I want to show you this protein here. Mascot says this is a hit, okay? And what Mascot says is that it's this protein from this organism. Now, that's a bit strange because I can tell you that we have never, ever worked on this organism in our lab, and there should not be any of this protein in the lab. But if we look at that, the score is 51. That's quite acceptable. The mass accuracy, the delta mass value here is very small. That's acceptable. All right, we haven't got very much coverage on the peptide, but it's a short peptide. And there's the data set. The mass errors are quite small. That data set is actually quite good. But I can tell you that that protein has never been in our lab. I'm going to have to stop sharing and go back. Sorry. Okay, I'm going to share screen to you again. Okay, you should see my screen again now. So this is where we were, and I'm telling you because I know that that protein has never been in our lab, so how can we have a hit for it? Well, what you have to pay attention to is this. It says same sets. If we click on this, we've got the same score. So we scored 51 for this protein. We've got the same score for trypsin. Now, of course, trypsin is going to be in our sample because we put it there because it's a tryptic digest. So trypsin is there, and if we look at that data set, the protein has a different sequence, but the peptide, this peptide, has the same sequence as the previous peptide I showed you. And this is the only, so we have 8 residues. And so these 8 residues in trypsin are identical to the 8 residues in the previous protein. And that's the only example I can give you, possibly the only example that exists of an 8 residue sequence which is identical to that in a protein where there is no phylogenetic relationship at all. That is purely a random match. And that's the only example of that I have ever seen. So it's not that the mass spec is wrong. The mass spec is absolutely correct. The mass spec identified the correct peptide. It just so happens that in this big Swissprot database, there are 2 peptides from unrelated proteins that are identical, but that's extremely rare. I'm going to have to stop sharing again.
So I have a quick question.
Yes.
You mentioned that Mascot can show the results in 2 different ways. One was kind of the family-based whatever
Yes.
than the original one. How does
Yes.
it decide which one to show us?
Okay, yes. So you can toggle between the 2. So we are now, this is the protein view. I don't really know how Mascot decides which one it wants to show you. It seems to be quite random, but it says not what you expected, peptide summary. We can click on that.
Gotcha, I see that.
It shows us the other method for looking at them.
Okay, perfect.
Okay, let's take a look at another data set now. Okay, I'm going to load data set 4 now because I want to show you something. So I'm going to load data set 4. And I'm going to start my search, and hopefully it's not going to take forever. Seems to be running a bit faster. So what you can do if you're operating an instrument, and this is what we do, we don't normally do manual Mascot searches. What we would do is program the instrument so that this search is embedded in the instrument method, and so as one sample finishes, it will run the Mascot search while the next sample is running. Because obviously this can take, if you've got a lot of samples to analyze, you wouldn't want to do it this way. Okay, so we are, okay, let me show you, so let me show you the peptide family. And what we have here is, so this is tubulin, and you can see we have the same set of peptides, so the same
Share
score.
your screen again, please.
Oh, sorry, I apologize. I keep on forgetting to do this. Yeah, just a minute. Can you see my screen now?
Yes, thank you.
Yep, okay. Yes, so we have the peptide summary, and we have good score. 128 is highly significant. We've got 2 high scoring peptides. You'll notice, I wonder if I can mouse over these. Yes, okay. This is what I wanted to show you before. This is quite useful because this is the data for individual peptides. So Mascot has, what it does is it takes that the data for that peptide and it matches it against every single tryptic peptide in the database. And every single tryptic peptide gets a score. And the top score is this 70.3. And then you can see that everything else is a significantly lower score. So 35.5, this is random sequence, it's junk, and everything else below it is junk. And you can see it's listed, is it the top 10 there? The 10th score is 14.6, which is basically nothing. And so everything in the database, apart from these top 2, have given really low scores, which means that it's complete junk. That 35.5, you might want to manually validate that, but the 70.3 is a very good score. Also notice that we allowed for oxidized methionine, and where it's underlined methionine in these 2 sequences, that's because the calculated mass includes plus 16 for the oxidation. But the value of the peptide family summary is in this. So we have, so what it's done is it says this is Enteroctopus dolphinii. We've never worked on that, and there's no reason to assume that that organism would be in our laboratory. And you can see there are dozens and dozens and dozens of hits with exactly the same score. And this is the weakness of Mascot, is Mascot, or this type of analysis, is Mascot has successfully identified the protein, but it has no idea which organism it's come from. And the problem with this type of display is that's the only thing it's found. It's only found tubulin in this display mode. So this is why we need the second display mode, the protein family display mode. We'll reformat that now. And now what we can see is, well, that's probably a single protein. It could be 2, but it's identified 2 different types of tubulin. This species is just random. This actually means nothing. All that it's telling you is that it's found a tubulin, and it's also found this weird protein that I said didn't exist. But what it's actually found, if you drill down into it, it's actually found trypsin, which is what we would expect. The 4th hit, let's have a look at that, because I'm not sure if that is real or not. So this is Xenopus sequence, elongation factor alpha. Not sure if we would expect that. So we've got a single peptide, so it's not very reliable. Delta mass is okay. The score is moderate. Let's take a look at it. Okay, so there are, most of the major peaks are represented. There's quite a lot of peaks that are not represented. So what do we have here? We have 1, 2, 3, 4, 5, 6. Okay, that's interesting. So I would accept 7 consecutive residues, 7 consecutive amino acid fragments for a hit. Here we only have 6. However, the last one is proline. And what happens is very often the sequence run is broken after a proline residue. So I would probably say, looking at the mass errors are acceptable. I would probably say that this is real. But I'm trying to give you a feel for doing this type of analysis, because one problem with using algorithms to assess your MSMS data is if you have a very high score, if the score is several hundred, you have no problem, you've got a hit, no question, it's massively statistically significant. If you have a low score of, say, below 15, it's definitely not a hit. But there's this gray area in the middle, and this is a perfect example of it. This gray area where we have an iron score of 34. Mascot has said it's significant, but it's absolutely borderline. And what we do in our lab is we manually validate this data. We have a look at it and we say, do we think this is real or not? But also you have to have a reason to believe, you know, if it's a completely random match to a random protein from a random organism, it probably isn't real. However, if it's something that you're expecting because it's an organism that you work on, it's a protein you've seen before, then that adds weight to it being a real sample, a real hit. So I think we have probably covered enough. Do we have any questions? Does anybody like me to go through anything again? Would you like to look at some of the other samples? We'll have to wind up in about 5 minutes. So any questions from anyone? Any requests from anyone?
I have a question. So whenever we put in the miscleavages, can you just kind of connect that to what it means on the actual data that we're seeing? I mean, does it relate to protein coverage or? No,
it doesn't. No, what it does is, I don't know, I've got an example I can show you. Can I share screens with you again and go back to this data set, if I can. No, that's the wrong place. I'm going to stop sharing again. Okay, I haven't really got an example to show you, not easily anyway. Let me show you this though. So I'm going to share screens with you again. Okay, hopefully you can all see this. So we're going back to this is the tubulin data, and you have a value here of M, and all of these are 0. And what that means is for these peptides, if you look at these peptides, there are no missed cleavages. So trypsin cleaves on the C-terminal side of lysine and methionine. And so you can see that all of these 3 peptides will end in an arginine. And this is where the cleavage happened after, in this case, a lysine, and these 2 after an arginine. So there are no internal arginines or lysines in the sequence because there are no missed cleavages. If you saw a peptide that had an internal arginine somewhere, then this missed cleavages would be 1, which means in order to generate that peptide, there has to be an arginine in the sequence which was not cleaved. And that happens quite frequently. I haven't got a data set to show you, unfortunately, but that can happen quite frequently. So you can get, you can generate peptides with 2 or 3 or 4 or more tryptic residues which are not cleaved.
That makes a lot more sense.
Sorry?
I said that makes a lot more sense. Thank you.
Yeah, yeah. Any more questions? Okay, it's 5 to 2. If there are no more questions, I'm going to wind up. I'd just like to say next week we will have the first of our guest lecturers. We'll have Holger Kramer from Imperial College. He will be talking about quantitative proteomics. He talked on seminar last year. It was very good. So I would encourage all of you to attend that if you can. And I will end the meeting there. Thank you very much for attending.